Okay, folks, if you don't mind, I think we're getting pretty close to 10 after, so I'm going to get started. Again, for those of you who have not gone to tutorials yet, I'm hoping that you'll check the announcements page. There was a change in the rooms assigned to you. It's not 54, it's 51A, 52, 51B, 51C, or 52. If any of you have any issues with not being able to show up on a particular day, you'll stay with the same TA, but you can discuss with your TA if you have to come at a different time. I'm going to switch gears a little bit. Actually, the themes are still related. We're still looking at inflammation. We're still looking at the obesity epidemic. We're just going to take a look at it in a slightly different way. I want to use the same principle. I don't want to just give you lectures based on what you might be able to find online or on textbooks. I also want to be able to take you through a paper, and I want you to understand that paper in terms of the microbiome. In biology right now, just like meta-inflammation is a really big topic, so is the microbiome. Your microbiome, and we'll talk about the official definition for what the microbiome is in a moment, your microbiome impacts you in terms of your biology just as much as your own DNA. It may not seem logical, it may not seem even plausible, but there's a growing body of evidence to suggest that your microbiome, whether it's on your skin or in your gut or in other places, actually plays a huge role in your biology. And I'm going to take you through a paper, not this week, but early next week, related to the microbiome. So we have one genome. Our genome, except for the epigenome, which we'll talk very briefly about, we can't really alter our genome. However, we can alter the genome of organisms that are living on us. And this gets into this idea that the microbiome has this flexibility, has this capacity to be rapidly changed depending on what sorts of things you do to this organism or groups of organisms that are living inside of you. And this has a profound impact on us, and because we're interested in human biology, we have to learn a little bit about this. Now, the microbiome that we're going to be spending the most time on is actually the internal microbiome, the one that you've been hearing a lot about, how your diet affects your microbiome, and how different types of exercise affects your microbiome. And that's internal. But to get there, I'm going to actually use the external microbiome and talk a little bit about how we do things, how we understand things externally before we get into the internal or intestinal microbiome in a little bit of time. This may surprise you. This actually, when I first read this paper, and it came out about a week and a half ago, and it came out kind of unsurprised, kind of by surprise. No one had really expected to see these types of results. If these results are true, and I'm going to leave that up to you to determine whether or not these results are true. Your microbiome, I'm going to convince you by the end of this module, is affected by your diet, how much sleep you get, as well as other factors that go into like how much stress you have, etc. If this paper is true, when you change your gut microbiome, after a certain period of time, you can't change it back. No matter what you do, no matter what you eat, no matter what happens, your microbiome is not going to have the flexibility that it once had. That's bad enough, that your microbiome is going to be irreparable. That is one aspect, but what this paper does is actually goes one step further. If your microbiome is damaged, your future generations, your offspring are also going to have a compromised microbiome. And again, it's shocking, it's surprising, it's unexpected, because you would think that you gain your microbiome independently of your parents, but it may be that having the right microbiome and the microbiome being affected by the fiber that exists in your diet might be really important in terms of future generations. It's not something that, as biologists, we think about, but it is something that is potentially, if this story is true, a really big factor in our biology. So this microbiome is a fairly new concept. It's now very common to hear about it in a number of different courses, whether it's in molecular genetics or if it's in a nutrition course. But at the time, 5 years ago, when people started looking and talking about the microbiome, there wasn't a lot that you as an individual would have heard a lot about. And the microbiome for me is going to be a really simple definition. So it's all of the different bacteria, viruses, and we also have to include all of the eukaryotic organisms like fungi and larger organisms that grow both in your body internally, inside your gut, as well as on the body. And we think that roughly it's on the order of about 10 to the 14th bacteria internally and about 10 to the 10th bacteria on your skin. And again, it's another paper that came out fairly recently saying that our numbers may not be right. That when you hear that the number of cells that you have inside you is more than the total number of cells that you have in your own body, that number itself may no longer be accurate, and we have to go back and revisit this. So we think now that our genome is not something that can be easily changed. Maybe the epigenome is one way to do so, but the microbiome, all of those organisms living inside our gut, we change that on a routine basis. If you lose a few days of sleep, your microbiome is different than when you were sleeping regularly. If you are changing your diet and you go from a high-fat diet to a diet that's high in fiber, you will change your microbiome and all of the genes that are associated with those different bacteria. So the microbiota, the living organisms, and the genome that is encoded by those microorganisms is known as the microbiome. So I'm going to take you through some of the microbiome that exists on the surface of your body. We are right now exposed to millions and millions of bacteria just by sitting in this room. And you as an individual are always encountering different types of bacteria, viruses, etc. Most of the time, it has very little direct impact on your biology. But on your skin, you have a lot of different types of bacteria, viruses, and in some instances, eukaryotes. Some of you will have seen on social media, for example, all of you have these little tiny face mites that live on your skin, that live on the hair on your skin, and these will all impact your health as well. So I want you to tell me a little bit about what you know. What is PNAS? So I mentioned a Gao and Blazer paper in 2007. PNAS, what does that mean for you? Or does it mean anything for you? It's the name of a journal. Do any of you know the name of this particular journal? Have any of you heard of the Proceedings of the National Academy of Sciences in the United States? This is one of the top-ranked journals. But I want you to take a look at the data. So the data is actually something that we're going to spend a little bit of time on. The data that they are reporting in one of our top-ranked journals in 2007 involves 6 individuals. That's it, 6 data points. 6 data points is enough to get you a paper in one of the best-ranked journals. And again, it's a fairly novel at the time, where we had 3 men and 3 women that were used in this study. And you can see the data at the top for 3 men and 3 women. And overall, you can see that there's a couple of interesting trends, and you do not have to memorize the ratios nor anything else. Why was this paper published in one of the best journals involving only 6 data points? 3 individuals, 3 men and 3 females. It's because it was actually a tour de force in terms of technology. By that time, we were able to do what is known as next generation sequencing. You could take, for example, in a very short period of time, small quantities of DNA and sequence extremely rapidly and derive the sequence information and therefore the types of bacteria that were growing on your skin. Overall, there were a couple of really interesting points, and you should be focusing more on the points here on the left rather than trying to memorize all of the numbers or ratios that are here on the right. If you took a look at both men and women, what this data actually said was that over 95% of the bacteria growing on either men or on women were in common. We have 3 major types or phyla of bacteria that grow on skin as determined by next generation sequencing. And these are the 3 different phyla that they were able to identify. And this actinobacteria and the firmicutes are 2 that we're going to come back to several times, as well as the proteobacteria. So these are growing on your skin. Doesn't matter if you're male or female, 95% of the bacteria that grows on you is identical to the bacteria growing on someone else's skin sitting right beside you. 75% of the total bacteria species, though, were found to be unique between individuals. And what this means is everyone has a slightly different proportion of the types of bacteria that are growing. These are phyla, right? Large classifications. But if you were to take a look overall, even though 95% of the bacteria are the same, that there are differences in proportions and the types of species of bacteria that grow within these phyla. So we have a lot of uniqueness, but we have a lot of commonality, which is what points 2 and 3 are trying to suggest. Now again, I'm not going to ask you to memorize this entire diagram, but what this actually shows is that when you take a look at your skin bacteria, yes, we have a lot of bacteria in common. We have 3 major types. 95% of everyone here has the same types of proteobacteria, actinobacteria, and firmicutes. Everyone here has that. The subspecies might be different, but it's also different depending on where you take the bacterial sample. So if you took the bacterial sample from parts of your face, it would be different in terms of the bacteria that are growing, for example, around your navel, or different between the bacterial species that exist on your foot. So different skin areas on the body, even though we have the same types of bacteria, from one individual, and this would be you, when we take samples from different parts of your body, we'll actually see different populations of bacteria. We have unique fingerprints of bacteria living on different parts of our body. And again, you don't need to memorize all of the different pie charts or diagrams here, nor do you have to memorize all of the different body parts from where these different samples were taken. And again, when you took a look, a separate study in 2007 showed that when you were to subclassify the different types of bacteria that you took from your skin, and these are highlighted for you because I do want you to memorize these, that one of the things that on your skin, irrespective of where you're taking them, whether you're taking them from your face or you're taking them from your arm or skin on your leg, etc., the most common types that you would find on you, on any individual, would be these types here: the Corynebacterium, the Propionibacterium, acnes, as well as other types like Streptococcus mitis, Feingoldea, etc. These were the most common types, irrespective of where you're looking on your own body. And again, there are differences in the skin bacteria between men and women, but overall we share the same phyla of bacteria. The other interesting thing is that over time, if nothing else happens to you, that if you are nice and healthy and you are living the same kind of life that you are living now as you will be when you're 60 years of age, the types of skin bacteria, the populations of bacteria, don't really change much over time. They change if you have a disease state, if there is a change in your stress levels, if there are changes in your diet, etc. But over time, there are very few changes in skin bacterial populations, as long as there isn't something really dramatic happening to you. So again, we're using this skin sort of as a model for what happens to us internally as well. So in order for us to talk about this in some more detail, I need to take a little bit of a detour, because I know in some of the courses that you have, you've learned a little bit of anatomy or you've learned a little bit of physiology, but I want to make sure that we're all on the same page. Your skin actually has a number of unique glands that are associated with it. These glands will directly influence and impact the types of bacteria that grow on these different types of tissue. So why, for example, might you have a different bacterial population? Because the skin itself might be a little bit different there. So we have 5 major gland types that are on different parts of your skin: the apocrine, the miracrine, ceruminous type, sebaceous. The mammary glands are ones that are included in this, but we're not going to be talking about them, and we'll only be concentrating on the top 4. But I wanted you to be aware that the mammary glands are actually considered as a gland type on your skin. So another way to say this is to talk about sudoriferous or sweat glands in terms of your body as well. We have 2 different types of sudoriferous or sweat glands, the miracrine that I'll talk to you about in a moment, as well as the apocrine. So these two different subtypes of sweat glands are a little different. For the most part, their secretions are identical. We have 99% water with a few salts tossed in as well. We have traces of metabolic waste. This is really important because this is one of the functions of sweat that we take for granted. Our sweat has a normal pH range of about 4 to about 6. And by doing so, sweat actually produces what is known as an acid mantle. It is a protective barrier. So when you think a little bit about this, why is it protective? Because these slightly acidic conditions that your sweat produces actually inhibits bacterial growth. Now, we have bacteria growing on us all the time, so it doesn't prevent their growth. It just makes sure that bacteria don't overgrow a particular area of your skin. And so this acid mantle produced by these two different types of sweat glands that I'll go into in some more detail in a moment are really important to us in terms of our biology. So the first type of sudoriferous gland actually has two names, just to keep them straight. These are known as merocrine, and they're also called eccrine glands. They're the first type of sudoriferous glands. These are the most numerous types of glands that we have throughout our skin. So they're the type of sweat glands that you have, for example, on the palms of your hands. They're the ones that are there on the soles of your feet, on your back, as well as on your chest. For the most part, it's where you have glabrous or smooth skin, in other words. And these eccrine glands, as I'll show you on the next diagram, are classified as simple epithelial tubular glands. And the epithelial cells themselves have a simple structure associated with them. They all secrete into a tube that I'll show you in the next slide. So these types of glands, even though 99% of it is water with some metabolic waste, a little bit of salt, etc., exactly the same as the apocrine type that I'll get to in a moment. These are really useful when your body needs to cool down, and they're great for maintaining the core body temperature. So again, they're simple epithelial tubes, but in addition to that, they also contain muscular epithelia. So they have myoepithelium, and the myoepithelium is capable of contracting just like any other muscle tissue. These are responsive to sympathetic signals from your nervous system. So when you're stressed out, when your sympathetic nervous system is active, that fight or flight response acts to contract these myoepithelial cells, and that's one of the reasons why when you're nervous, you tend to sweat. These are the glands that are being activated, and it moves that sweat up from the glands up to the surface of your skin along those tubes as these myoepithelial cells start to contract. And this is that merocrine or eccrine gland that I just mentioned to you. And surrounding the outside of these eccrine or merocrine glands, we have these myoepithelial cells. They contract. When they contract, they squeeze the fluid up along the different types of tubes or duct systems that you see here, and it moves to the surface of your skin, which allows for perspiration to aid in cooling down your body temperature. Or it might be activated when you are feeling really nervous or really excited, or if you're feeling really frightened and your sympathetic nervous system is active. The other type that is also shown here is slightly different in how it looks. This is the apocrine sweat gland. It's often associated with this hair follicle that you see here, and its secretions generally follow along with the hair follicle itself. So again, I'm not going to go through this in a lot of detail, but just to give you an idea, because we'll need to talk about this in some more detail later, your skin has these three basic layers: the epidermal layer, the dermal layer that you see here, as well as the subcutaneous layer that you see all the way down here. And associated with the subcutaneous layer, these structures here is fat. The types of adipocytes that we looked at earlier, the adipocytes sit down here as well. So I often ask this question, and it's useful for you to maybe jot this down. They're found in different locations. So not only are they structurally different, but you can see that the subcutaneous layer is where we would find the beginnings of the apocrine gland, and the dermal layer is where we would find structures that are normally associated with the miracrine gland up here at the top. And again, the subcutaneous layer also contains all this fat that you have that contains those adipocytes that we looked at earlier. So this last type that I mentioned to you, the apocrine gland, is the second type of sudoriferous or sweat gland that I mentioned to you at the outset. So sudoriferous glands, two different branches. One is the miracrine ecrine, the other one being the apocrine gland. These are not found on smooth skin. These are concentrated in places where you have a little bit more hair, like under the arm, the axilla. Your axillary glands are your armpits, your groin areas, and other areas that tend to have more hair. So these are the ducts that are associated with the hair follicles, and the sweat produced is, again, very similar but not identical to the miracrine type. It's not that clear type of fluid that you get. It tends to be a little bit milkier. It's a not as transparent, in other words, if you were to collect it in bulk, and that's primarily because it also contains fatty acids as well as proteins. Now, your bacteria live on your skin. Your bacteria need things to be able to reproduce. They need to digest and take in different compounds. And one of the reasons why we have different bacteria growing on different parts of your skin is your sweat glands produce different elements that are associated with it. And in particular, these apocrine glands have these fatty acids as well as proteins. These apocrine glands are also things that develop a little bit later. And that's one of the reasons why I mentioned at the outset that overall, throughout your adult life, your skin microbiome, or the microbiota, the bacteria in particular, tends to be pretty stable. But during puberty, we know that there is a slight change in the microbiome. And one of the reasons why is these apocrine glands tend to develop just a little bit later during that pubertal stage. So this is also the type of gland that produces bromhydrosis. And again, if you ever play sports, or if you ever walk through the athletic center and you've gone to the change room kind of smells, right? Or you've gone over to Heart House, the change room smell because the sweat here in particular tends to smell because the bacteria break down those fatty acids which produce that characteristic sweaty odor. And this is bromhydrosis, the odor associated with the breakdown by bacteria of your sweat. Okay, so the last gland the second last gland that I'll mention to you is the ceruminous glands. And these are ones that we don't find very commonly. These are only found in the external ear canal, and their secretions are a little different. It's not watery. It tends to be more waxy in terms of its overall appearance. And one of the reasons why it's more waxy is because they produce this substance, just like you would suggest, as the name suggests, this sebum, which is oily. It's an oily gland substance. And in addition to that, this oil-like substance mixes with the skin epithelia to produce this earwax. And this earwax is actually really important because as some of you will know, in the summertime, especially when you get bugs flying around, it actually prevents bugs while you're sleeping from crawling into your ear, right? And one of the reasons why this ceruminous substance is important is because it tastes bitter. Don't ask me how people know that it tastes bitter. You're just going to have to take my sort of slide for granted, and it tastes bitter, and that bitter taste actually helps to repel any insects from going into your ear. It's also important because you don't want bacterial growth. This is the external ear canal. If you ever have bacteria growing inside your middle ear or into your inner ear, it actually will cause a number of physiological problems, which you don't want to have because it's really hard for you to fix those types of problems. So this helps to inhibit any type of bacterial growth, and in addition, it prevents things from getting into your ear by keeping the outer ear hairs a little bit sticky. So the last type are these sebaceous glands. And these sebaceous glands, again, are over the entire body, not on the palms, not on the soles. This is also an oily substance, much like the ceruminous glands. We don't have dead cells to go along with this, but here the entire cell itself breaks up. And when the cell breaks up, the individual components are combined with the substances that are secreted to form the secretion, or a holocrine-like secretion, which is being secreted from these types of glands. So again, just like we saw earlier on, many of these are associated also with the hair follicles, very similar to those apocrine glands, in other words. And the function of this type of sebum as well is to also collect dirt, much like the ceruminous glands, also to collect dirt and to help lubricate and soften the hair as well as the skin. So if you have really dry skin, or you don't have really long, luscious, flowing hair, you need to work a little bit on your sebaceous glands and get them stimulated to work a little bit. And then you'll have really great hair. And you can tell that I put a lot of effort into my own sort of sebaceous glands on the top of my head. So why is all of this important? So some of the sebum that I mentioned is found throughout the entire body. So why is the sebum important? So if you take a look at the sebum that is produced by these sebaceous glands by the seashore, and sorry, I had to put that in there. If you take a look at all of this, one of the things that I mentioned to you earlier, that 2007 study, one of the types of bacteria that they found was this Propioniae bacterium, and the subtype being the P. acnes that will be coming back to you several times. P. acnes does not just grow on your face, it grows on multiple parts of your body, as we'll see on Friday's lecture as well. So this is a gram-positive rod. It is also called a bacillus. So any type of bacillus is a rod. It's a type of shape. Do you know any other types of bacteria? So we have rods, which is what you're seeing here, these little rods that you see there. And then we have these other types sort of interspersed, these little dots. What do you call those little dots? Those of you who've taken other microbiology courses or genetics courses. Yes. A coccus, that's right, or cocci for multiple ones. So if you hear streptococcus, that's a type of bacteria which is round in shape when you do a gram staining. When you hear staphylococcus, it means it's a round shape as indicated by gram staining. So the one that I want you to make sure that you know is this P. acnes, or Propionibacterium acnes, and it is a rod, it is a bacillus. And this bacteria that grows normally on your skin is used to digest that sebum that I mentioned to you earlier, as well as the keratin that's found on your dead skin cells. Your skin cells contain lots of this cytoskeletal protein keratin, and as they flake off, like every time you sort of scratch your skin, you'll see that you are picking up this dead skin, and when you do so, that contains a lot of this keratin that is eaten by this P. acnes that normally is found on your skin. So what happens normally is that if, for whatever reasons, the bacteria that is normally eating that starts to multiply, one of the things that happens is that it produces an inflammatory response, and it draws in through that inflammatory response these neutrophils. And these neutrophils, especially if they're trapped in the skin, or the bacteria is trapped in the skin, will start to digest the bacteria, and as a result, it will produce this digestive pus or pus pocket from this. And these are those whiteheads that some of you might have had a long time ago when you were in high school, and that's because the bacteria has proliferated. It has drawn in these neutrophils, and the neutrophils have digested those bacilli and produced these little whiteheads that contain these pus pockets. So this P. acnes is actually a very, very common type of bacteria. It grows all over your skin. It's not just on your face, not just on your neck, etc. It's all over your skin. And in fact, if some of you take upper year courses related to general surgery, or you take courses related to human biology in your fourth year seminars, one of the things that is surprising to most students, but perhaps shouldn't be, is that if you were to go in for a surgery, if you were to go in for a joint replacement, or you were to go in for a cardiac shunt, or you were to go in for a replacement heart valve, where it's fairly invasive. You're going into other organ systems, you're cutting through the skin. One of the things that is a very important post-operative opportunistic, it takes use of that chance or the opportunity provided to it, is this P. acnes. And this is one of the most common type of skin disease that infiltrates inside after a post-surgical site infection. So these are dangerous. When you have gone in for an operation, they've cut open your skin, and for whatever reasons, your immune system is not working well, or they have improperly sterilized that area. These are the types of post-surgical infections, including P. acnes, that will directly impact your recovery. So there is no evidence right now to suggest that diet alone will affect your acne. My kid is 16, and so she worries a lot about how she looks and whether or not her skin is clear and whether or not she should be wearing makeup or eating oily foods. There is no evidence in the literature to suggest that if you eat a lot of oily food, that you're going to have more acne than someone who doesn't eat oily food. But overall, it becomes a big issue. Why is your skin more oily? And how do we take care of this? Because it is a big issue for a lot of individuals. They get really anxious about the way they look, and then it impacts on how they deal with other individuals and the quality of life that they have overall. I can tell you that for me, stress really impacts like my skin a lot. And I'm into my 40s, and if I'm like really stressed out, I still have breakouts of acne once in a while as well. Our strategy, by the way, is not for taking care of acne, is actually fairly common. What we try to do is get rid of the bacteria, right? It makes a lot of sense that it would make our lives easier just to get rid of the bacteria that grows on your skin. So one way is to use benzoyl peroxide to dry up any of those sebaceous glands, to remove any of the sebaceous glands that are plugging up the follicles, and then kill off any of the bacteria that are living there. We could use antibiotics. We could use antibiotics like tetracycline, and these are often used in treatment as well. Or we could use things like Accutane or isoretinoid, which is only available by prescription, which can also help to eliminate these. So we use antibiotics, really powerful antibiotics to do this. Again, they're bacteria, why not use antibiotics? The problem is right now, we use antibiotics all the time. And these antibiotics are becoming less and less effective, and actually they don't really do the rest of your body any good. And we'll talk about that in more detail on Friday. So again, these acnes this isoform is not only found on your skin, these are found in your oral cavity, they're also found in your large intestine. You'll have to think about why it's found inside your gut as well. And hopefully by the end of Friday's lecture, you'll understand why. It's found on the conjunctiva of your eye, as well as in the external ear canal. It's found everywhere, in other words. But here's this thing. Right now, if you were to take something like Accutane or isoretinoid, or you take tetracycline, which is a very old antibiotic, and it works, it does work if you're doing it by prescription, it's actually not great for you in terms of your biology. And so people know that there are lots of side effects. For taking isoretinoids, you need a prescription. You need a prescription not just because it's an antibiotic and we need prescriptions for antibiotics in Canada, it has a number of side effects that are associated with it. These drugs that you use, like antibiotics, come at a cost. So these microbes actually are really important, and we think if we use those similar principles that we talked about earlier, you have bacteria growing on your skin all over the place all the time. Something happens to cause the bacteria on your face to suddenly start proliferating like crazy. Maybe it's stress. I don't think it's diet, but maybe some people think it's diet. Maybe there are other things going on. Suddenly you get a lot of acne where follicles are plugged, and then you have neutrophils coming in to cause that redness and eventually those whiteheads. But another idea that is fairly recent is if we have these different bacteria growing on your skin, why not use them to control the growth of other types of bacteria? So it turns out that when you look at the human skin and you look at all the genes expressed by the different types of bacteria that are associated with the bacteria that form acne, we actually can subclassify these into different types of strains. So up until like 2007, by the way, where you see this massive proliferation of papers related to the microbiome. Before that, we knew about the microbiome, but it was really difficult to do experiments related to the microbiome. We didn't have the technology. We just simply could not do the types of experiments. Like, ideally, right now, I'd love to be able to give you a whole lecture as a hologram. Sitting in my office, I could just have a hologram up here. We don't have the technology. We dream about being able to do things, and that's what we were stuck at in 2007. We wanted to do all these things like genome sequencing. We just didn't have a way to do it. But in 2005, 2006, that technology became incredibly efficient and allowed us to do these types of sequencing that I'll talk about throughout the rest of this course. So I just told you that this P. acnes is important for different types of acne breakouts, but did you ever have that friend in high school? I had one friend in high school. Like all my other friends, we always had these breakouts. We'd have like Clearasil on our faces when we walked into class. You could smell Clearasil when you walked into class. But you ever have that one friend who had like perfectly clear skin throughout high school? Beautiful complexion all the time. It turns out it's not unique. There's a small proportion of individuals, about 15% of teenagers, for example, will never have a breakout. 85% will, but 15% will not have a breakout. Adults also tend not to have breakouts as frequently as they get older. So why is that the case? Why do some teenagers never get it, and why does it change when you become an adult? And as I just mentioned to you, our genome, like the types of bacteria growing on our skin, don't really change with age. What is accounting for this type of difference? So we didn't have a clear idea until these genomic approaches became feasible in 2006 and 2007. And one of the things that this paper that I'll take you through really briefly by Fitzgibbon and Lee did was they wanted to look at the bacterial profiles from people that never had breakouts as teenagers and people that have really bad skin. And they wanted to know, what's the difference between the type that have perfectly clear skin versus the type that have lots and lots of bacterial breakouts. And one of the things that they did was to use healthy skin microcomedomes versus the comedomes that came from individuals that had pimples. Do you know what a comedome is, or microcomedome? Yes? Have you heard of it before? How many of you snore at night? How many of you wear those little strips to prevent you from snoring? No one. Okay, that's okay. How many of you have ever used those little strips that you put on your nose to help you breathe better, like you're a pro athlete or you're a high performance athlete? Have you ever taken it off after? You must have. You're not wearing it now, right? Okay, so you take it off after, and what do you do with it? Throw it out. That's the logical response. Thank you. So if you're a biologist, though, before you throw it out, you take a look at it, right? And if you take a look at it, you see these little things that have come out of your skin, out of the little pores that are in your skin. And those little pore fillings are these comedomes. And these comedomes contain lots and lots of different bacteria that you can then sequence. I almost feel like we should do that as sort of a homework exercise. Everyone go out, get those strips, and rip it off, and then show me what comes out of your skin. And then using this type of sequencing data from these different comedomes, it was found that there were about 66 different subtypes of bacteria that existed within the different pore plugs that came out of the skin strips. But about 90% of the bacteria were P. acnes. Okay, we had lots of different types, 66 different ones, but 90% of what they saw was actually due to P. acnes. And again, P. acnes we can actually subdivide into subtypes or subgroups. Two of these subtypes of P. acnes were found to dominate on pimply skin. We found one P. acnes type that dominated only on pimply skin, but was not ever found on normal healthy skin. So something changed. Pimply skin, you got this type to grow. And this third subtype was only found on healthy skin and not on pimply skin. So if you didn't like using different types of drugs, what might you try to do? You would try to get this healthy type that you only see on healthy skin growing on an individual, because it wouldn't be involved in attracting neutrophils. It wouldn't be involved in growing and plugging pores on your skin. And so this is sort of a new strategy. Can we replace bad bacterial types, these two subtypes that were dominant on pimply skin? Can we actually repair your skin, for example, by getting a different bacterial subtype involved? And this is sort of a new strategy that individuals started to look at. And again, you don't have to worry about memorizing details from this part of the slide. If you're really interested on comedomes and how these things work, you can read up on it. I'm more interested in the bullet points that are here for you on the right. So this is sort of a theme that we're now going to be looking at. So we can examine things on the surface of our skin fairly easily. We have access to things on the surface of our skin really easily. We have bacteria growing on the surface of our skin that we can access easily. But this is the same idea that we're going to apply internally to our gut. So can we actually sample things in our gut in real time? Can we influence and impact things, just like with seeing breakouts on your skin with acne? Can we take and apply the same principles but only internally inside our gut? So we're going to take a look at this. We're going to see that this colonization, just like with the skin on our surface, is not going to be uniform. It's not homogeneous. And we're going to see that it can also change, but also stay constant throughout your life if you're healthy. And the other thing is, just like that last Lee and Fitzgibbon paper that I showed you, in theory now, and this is what we're actually really hoping for, because we know that antibiotics, if you've been reading the various newspapers or news articles and journal articles, we know that bacteria themselves are developing different types of resistance to the traditional antibiotics that we had. So if you had, and we'll talk about this on Monday, not this coming Friday, but on Monday, if you had a Clostridium difficile infection right now, the traditional approaches to getting rid of that infection don't work anymore. They're no longer effective like they were 30 years ago. And so what they have to do now is use a new approach of seeding your gut in a different way. So we're going to look at how we control our gut microbiome. And if that paper that I showed you at the outset is actually correct, there's only one paper out there in the entire universe right now, as far as we know, that says that your gut, if you stop eating fiber, is changing your gut microbiome, and you reach a point where you can no longer reverse that, and that irreversibility is passed on to your children, and they also can't go back either, that over time we are changing as a species our gut microbiome. If that paper is correct, and if that paper in mice actually applies to the human condition. So we're going to see that lifestyle affects your microbiome a lot. We're also going to see that there are early developmental changes. So when you are born, your gut microbiome starts a certain way. And then when you reach adulthood, it becomes really stable. And then as you reach an elderly status, it might change again. And we're going to see that some of the ratios of different bacteria are going to change. And again, right now, you don't have to worry about memorizing this particular diagram. I'll take you through different elements of it. But whether you take antibiotics, whether you exercise, the types of food that you eat, all of those factors impact on your microbiome. Different types of infections that you'll encounter will impact your microbiome. So there are many different types of things that are going to impact your microbiome. So whether or not you wash your hands affects your microbiome. Some of you, for example, because you have not washed your hands regularly and you've come in contact with individuals that also have like a Helicobacter pylori infection, are going to be infected with Helicobacter pylori in your stomach. Will you know that you have the infection? Not at all. Not until you are into your 40s and you have problems with your digestion. We know that, again, diet plays a huge role. Whether you are actively engaged in exercise will also play play a huge role on the types of microorganisms living in your gut. If you take antibiotics, when you take those to cure your acne, it also will impact other parts of your intestinal microbiome. And whether or not you are meta-inflamed, whether or not you have type 2 diabetes, type 3 diabetes, etc., will also impact this as well. And all of these things may actually play a role in your metabolic status. Okay, and this gets back to what we talked about last week. So I need to think a little bit about this. How are we going to impact your microbiome? And how do we change our microbiome? Is it really that easy to change your microbiome? How many of you think that it's really easy to change your microbiome? No one? How about if you took antibiotics for a full week? Would you change your gut microbiome? Yes? Okay, so you can change your gut microbiome. Do you think that your gut microbiome is going to change based on what you eat? Yes? Okay, so you know, one of the funniest questions I was asked at the start of class today, what do you have for us to eat today? Um, so yes, this course is a little bit about gluttony and eating. And yes, I have stuff for you to eat. Do I have any volunteers? Yeah, a couple of you. Um, Actually, this is a really trick question, and this was actually really bad. So your cafeteria, because I didn't have a chance to run over to Metro today, your cafeteria is doing a great job of ripping you off. That's all I can say. I paid for all this stuff, and they gave me the bill, and I said, really? It's like 20 bucks for all of this, and it was quite impressive. So you have this core power drink. I don't know why they're selling core power drinks over in the cafeteria, yet they are. You have this, yeah, you have this yogurt drink, I guess, which some of you might think is good. And then you have this Greek yogurt. And then my favorite is actually something that I would actually highly recommend for some of you. This, your Awake bar. Which of these do you think is the least healthy? In terms of what I told you earlier on. Any ideas? Is it that one? Actually, it's not bad. That has 1 gram of fiber. I don't know where the fiber comes from, by the way, but it has 1 gram of fiber. So it turns out, yes, at the back. The yogurt drink, you're right, no fiber, lots and lots. There's no active culture, by the way, in this. So it turns out actually to be the least sort of healthy option for you. So you guys decide among yourselves what you're going to eat today.